Comienzo. Muy buenos días. Los saluda el doctor Eduardo Gotuso de la Universidad Peruana Cayetano Heredia. Tengo el gusto y el honor de moderar esta sesión. Esta sesión se ha organizado con GVN, que es una alianza estratégica internacional que relaciona todos los grupos que hacen investigación en virología. El doctor Christian Bresco de Francia preside esta asociación, esta red internacional, en conjunto con su fundador, el doctor Robert Gallo, que también participará en los siguientes días en nuestro congreso. Hoy día, por razones técnicas, todavía el doctor Christian Bresco no se está enrolando, pero yo quisiera señalar que GVN invita a los virólogos de América Latina a ver la página web, a incluirse como grupos de investigación. Son grupos muy distinguidos de todo el mundo, tanto del Asia como de África, de América Latina, de Europa y de Norteamérica. Todos están participando, incluso grupos de Australia. Y entonces quisiéramos invitarlos a ustedes a reconocer esta alianza internacional tan importante de crecimiento. Hoy día tenemos 4 charlas y me voy a permitir presentar a cada uno de ellos. La primera charla está el doctor Guido Poli. Doctor Poli es un inmunólogo, virólogo, es especialista en oncología en la Universidad de Ferrara en Italia. Es jefe de la unidad de inmunopatogenia de SIDA en el Hospital San Raffaele de Milano. Es profesor de patología de San Rafael en Milán. Él recibió recientemente un premio a la investigación clínica de la Sociedad Europea de Investigación Clínica y coordinador del doctorado de Oncología e Inmunología Aplicada Básica de Medicina Nuclear. Dr. Guido Poli, please, I don't know if you are ready, but I introduce you, your summary about your talks, and finally the title of the lecture is Role of the Macrophage in Respiratory Syndrome, and the reservoir about the absence of CRT. Dr. Poli, welcome to the Pan-American Infection Disease meeting.
Thank you, Dr. Gotuso. Can you hear me well?
Welcome.
Okay, thank you very much. Okay. Thank you very much for your kind introduction. Would have loved to be with you in presence, perhaps the next time. Can you confirm that you can see my presentation, my first page of the presentation, before I move on? Can you see it?
Yes, we are seeing the slides.
Okay. Okay, great. So this will be, as far as I understand from the program, the only presentation dedicated to HIV infection and AIDS, whereas the other will be dedicated to the COVID-19. And I will deal in about 20 minutes with the issue of macrophages and viral reservoir in the presence and absence of the combination antiretroviral therapy. So just as a way of a little bit of background, I think most of you will know that the discovery of a combination antiretroviral therapy, CR, has completely reverted the fate of infected people. We all know that before ART, people was essentially condemned to die in most cases from AIDS. So since the mid-90s, 1996 has been the very first breakthrough paper, the combination of different drugs targeting different points in the HIV life cycle has significantly improved, has blocked viral replication, and essentially gave back life to people who otherwise were condemned to die. However, there is one major limitation of combination antiretroviral therapy, which is that no matter how long you maintain antiretroviral therapy, no matter how intensification you can make. If you stop antiretroviral therapy in more than 90% of individuals, the virus will restart to replicate again, and disease progression will restart again. And so on the one hand, this means that infected people are essentially have to take antiretroviral therapy for the rest of their life, with significant cost also in terms of public health. But from a biological and pathogenetic point of view, the question is where the virus hides. And in the scientific community in the last 15 years at least, has come up to the conclusion that the major reason why antiretroviral therapy cannot eradicate or purge the infection from an individual is because HIV as a retrovirus can establish one or more reservoir of cells that are latently infected or persistently infected. I will elaborate further on this concept, carrying replication-competent integrated HIV proviruses that upon suspension of CR, they will rekindle the reprisal of the infection. And a lot of elegant work from Robert Sidiciano and other groups have essentially identified in a subset of resting memory CD40 lymphocytes a major component of the so-called reservoir. A relatively small pool, the numbers here have actually grown over the years, so the pool is not as small as it was thought in the beginning, but essentially a small pool of cells that become infected but do not die upon infection. They carry on integrated proviruses. And they maintain themselves through a variety of mechanisms, including homeostatic proliferation. And essentially, it is these cells that in the moment in which a patient suspends the antiretroviral therapy assumption, they will restart the infection. And this is a very well-established fact, the role of this reservoir. Then, just going back a few years ago, in 2014, there was another breakthrough observation, which is the fact that there is expansion of clones of infected cells all carry on the same integrated proviruses. So this is not like neoplastic transformation, but it's something in between, because the virus can integrate in specific sites in the human genome, including proto-oncogenes and genes that are expanded and that are expressed particularly in certain subset of T cells. And therefore, as you can see, up to 40 or 50% of the clonally expanded cells have carry-on viruses, proviruses with that can be part of their inducible reservoir. And some of these proviruses are indeed made of replication-competent virus, as later demonstrated. So this great advancement has led to a sort of what I would call a little bit of a biased view of the reservoir, in the sense that there is what I would call a CD40 cell-centric model. And I will explain what I mean by fractal view of the HIV reservoir. So essentially, a lot in the scientific community say, well, now that we have established that CD40 cells are the reservoir, perhaps all the effort should be dedicated to understand on how to suppress or eliminate CD4 cells. So essentially, it's like considering the reservoir a monolithic, a CD40 cell-centric monolithic fraction. But before offering you a little bit of a different perspective, I would like to just make a point about what we could call a semantic, but actually a very relevant reality. We should come on to agree, or at least consider, that the terms HIV latency or proviral latency and HIV persistence are very equal concept. In other words, is viral persistency exclusively explained by proviral latency in CD40 cell or other cell types? Or is viral persistence include, of course, proviral latency, but also other additional forms of scientific evidence essentially suggests that these additional candidate cells to the reservoir could be myeloid dendritic cells, which play a very important role in triggering the primary immune response, the follicular dendritic cell in the lymph node germinal centers, astrocyte in the central nervous system, and, as I will discuss throughout the remainder of my talk, the tissue macrophages, which are ubiquitous cells. So even if you're not a specialist, you immediately realize that if the reservoir is indeed contributed by a variety of cell type, well, that implies that not all approaches, not all therapeutic or strategies may necessarily be efficient enough for all these cell type, and that we may need to think of different modalities to interact with different cell type. So let me remind, before we move on, of the fact that the HIV infection of macrophages, particularly in the brain, but not exclusively in the brain, has been recognized since the very early days. You can see these papers are from 1986, so very early days, in which essentially the virus was visualized. I mean, viral infection itself was visualized in the brain of people with AIDS encephalopathy. And actually, let me remind you also the fact that the encephalitis caused by HIV and we came up with a first consensus review on the concept of HIV reservoir and cure. And just a little extract in this cure, we say basically the concept I already told you, that in addition to CD4 T cells, other cell types should be regarded, and you can see myeloid cells, meaning essentially macrophages, should be strictly considered as candidate to be relevant for HIV infection, resistant to the purging of combination antiretroviral therapy. And there is a number of evidence that this is really the case. For example, in this paper in 2014 by the group of Mirko Paiardini and Guido Silvestri, they look at CD4 T cell depletion in HIV infected macaque. And what they saw is that by eliminating the CD4 T cell, that if you think about is the picture of AIDS people with no CD4 T cell around, essentially the virus jumps, so to speak, to macrophages and macroglial cells. And they make a very great contribution to maintaining virus replication in these experimental infected animals, as also clinically seen in AIDS patients with no CD4 T cell. And let me just remind you, without reading the slide, that HIV belongs to the subfamily of lentiviruses, in which there are essentially which are viruses that are particularly tropic for myeloid cell and macrophages. The difference for HIV is that by picking up CD4 as a primary entry receptor, it expanded its tropism to the CD4 T cell, giving rise to the AIDS as we know it now. So as I mentioned to you, there is a discussion in the scientific community about whether macrophages are a relevant or not a relevant cell type to be considered for dealing with the issue of viral reservoir. So the skepticism, the main point of the skepticism is that for the case of CD4 T cell, as I summarized, there is a robust now evidence that they are long-lived due to homeostatic proliferation and other mechanisms, such as expansion of clonal T cell. There is some evidence that the macrophage can phagocytose infected CD4 T cell, providing a sort of false positive signal in terms of infection, and that by certain analysis of tissue of infected individuals or animals, the number of contaminant T cell in a certain portion of tissue could explain a positive signal attributed to the tissue macrophages. Well, to these 3 points, I would like to offer 3 counterpoints for the literature, and I will briefly elaborate further. And the first of all is that there has been what I like to call a Copernican revolution in macrophage biology in understanding their nature. And now it is clear that what we call now tissue-resident macrophages are long-lived cells capable of homeostatic proliferation, exactly like resting memory CD4 T cells. And the number of studies, I will summarize just a couple of slides, in non-human primates and humanized mice have demonstrated infection of tissue macrophages in the absence of contaminant T cell. And I find that at the end of my talk, I will talk to you about a specific feature, which is the capacity of macrophage to accumulate infectious virus in the so-called virus-containing compartment, which could represent a sort of a different kind of reservoir. So, first point is that we, in the last 10 years approximately, we realized that bone marrow independent origin of tissue-resident macrophages exist in the embryonal life and colonize our tissue, and due to homeostatic proliferation, they remain through our lifetime. So these cells are not replenished by bone marrow dendritic cells, whereas bone marrow-derived monocytes can be recruited in the tissue in response to chemotactic signal in the case of infection or inflammation. Then, as I mentioned already, number of studies in relevant animal models excluded the necessity of so-called contaminant T cell to observe infection and latent, reversible latency in macrophages. The first paper in 2017 was published by the group of Victor Garcia in humanized myeloid-only mice, which about 33% of animals were capable of showing rebounding virus after treatment with antiretroviral therapy. And the consortium on SIV, similar immunodeficiency virus infected macaque, led by Janice Clement, essentially demonstrated the same thing, that indeed you can have infection of macrophages in the brain, and that even in the present. And this is not just animal model, because it occurs in humans too. And you can study, for example, by imaging studies, which a significant number of infected individuals under antiretroviral therapy show white matter alteration and signs of HIV infection or HIV-related inflammation. So giving the support to an editorial comment by Spector and Rappaport to say, if you ignore CNS, you do it at your patient level. So in the final part of my presentation, I would like to briefly talk to you about this specific feature of macrophages, which is the capacity to accumulate virions in so-called virus-containing compartments of VCC. And this is a dynamic reservoir, in contrast to the latent reservoir. Well, personally, during my 7-year stage in Anthony Fauci's lab, we describe this VCC in a surrogate model of macrophage infection represented by one cell line differentiated by Paul ester. You can appreciate the fact that in this huge cytoplasmic compartment expanding, find a lot of virions. And our group, as well as others through the years, has characterized a variety of very heterogeneous stimuli. You can see some of the stimuli here that can actually favor the retention of HIV production in this part. So one question that we asked was whether there were additional signals that could induce the rapid discharge, the rapid release of such virions. We identified the extracellular ATP engaging the P2X7 receptor, which is a very well-known damage associated with pattern feeded inflammatory reaction, is capable of rapidly discharging virions without killing the cell. So we now know that this reservoir is very dynamic, can be influenced in terms of viral accumulation or virion. And obviously the question is this just a fascinating artifact in vitro infection of macrophages, or are there evidence that this can be occurring also in vivo? And to my knowledge, the best evidence this is actually occurring in individuals receiving antiretroviral therapy came from Morgan Bonsello in Paris, where she studied the vitro macrophages of people undergoing gender reassortment. And what they found that these people were under suppressive retroviral therapy. We found indeed the macrophage associated HIV containing virus containing compartment, whereas the CD40 cell in the vitro did not contain either viral DNA or RNA. So, in summary and conclusion for this talk, what I'd like to summarize is the fact that the combination antiretroviral therapy blocks HIV replication and significantly restore the system of infection control, but cannot eliminate or suppress the viral reservoir, which is present in resting memory CD40 cell, but also in myeloid cells, such as macrophages. The tissue-resident macrophages are long-lived cells capable of homeostatic proliferation and resistant to the cytopathic effect of HIV, therefore qualifying as an additional reservoir which is unaffected by CR. And the final thought is that the role of macrophage-associated VCC deserves further studies aiming at purging or silencing the definitely integrated provider capable of producing infectious virus whenever CR is stopped. And in my way to salute you, I would like to let you know that we have a very elegant book coming out in press this year, which I edited with Elisa Vicente and Fabio Romelio, and lots of very prominent authors, which will be totally dedicated to the HIV reservoir, their modeling, quantification, and approaches to cure. And thank you very much, and good continuation of your beautiful meeting.
¿Pudieran conectar a la doctora Penny Moore, por favor? Dr. Penny Moore. Meantime, I invite again to our attendings about this lecture. Quiero volver a introducir GVN es una international network bastante importante. One second, it's only technical issue. They will right now present.
Good morning, good afternoon, good evening. Thank you for the invitation to present today. I'm going to show you some of the work that we've been doing looking at neutralizing responses against SARS-CoV-2 variants and try to summarize the implications for vaccine design. I'm based at the University of the Witwatersrand in South Africa, also at the National Institute for Communicable Diseases of the National Health Laboratory Service, and I have a joint appointment at the CAPRISA. So as I'm sure everybody who's been following the COVID pandemic knows, many monoclonal antibodies have been very rapidly isolated from patients who are infected with SARS-CoV-2. And some of these are shown here in this paper that beautifully summarizes their epitopes from Chris Barnes and Pamela Bjorkman's groups. You can see that there are many antibodies here and that they generally recognize 4 somewhat conserved epitopes on the SARS-CoV-2 spike, right up at the top of the receptor binding domain. And I think it's important to point out that all of these monoclonal antibodies that we are using both for therapeutics and for, in some cases, for vaccine design, were isolated from ancestral strains early in the pandemic. These monoclonal antibodies, however, all rely on residues that vary in variants of concern or variants of interest. And that's shown here from this beautiful review from David Corti on the left-hand side. You can see here a series of monoclonal antibodies, and their footprints are shown on the sequence of the spike of the SARS-CoV-2 variant in green. And what you can see in red are the boxes that identify mutations in the spike that are associated with variants of concern. So, for example, we're familiar with the 484 mutation, which emerges again and again in multiple variants of concern. And so many of these monoclonal antibodies show substantially reduced efficacy or complete lack of activity against variants of concern. And of course, the reason this is a problem potentially is that variants of concern now dominate the pandemic. As you can see at the bottom right-hand side, variants of concern are now responsible for almost all infections across the globe. This is certainly true in South Africa, as in many other countries, but we focused in South Africa mostly on the epidemic that has occurred in our country. And I'm going to show you some of the work that's come out of after that. In South Africa, we experienced 3 waves. We're currently, we hope, towards the tail end of the 3rd wave. The first wave was dominated by the original variant, and that's shown here in grey. The second wave, which occurred somewhere towards the end of 2020 and into 2021, was dominated entirely by a variant called, at the time, 501YV2 and subsequently renamed Beta. And you can see that on this graph down here. You can see epi-weeks shown here at the bottom. Where my mouse is now is the beginning of 2021. And you can see that almost all infections from that time onwards were caused by the Beta variant. We were interested right at the beginning of the emergence of the Beta variant in understanding what these mutations were that defined Beta, and the structural analysis very clearly indicated that these were likely to be immune escape mutations. And indeed, that has turned out to be the case, and there are now several studies that have confirmed that. I'm going to show you our data, which was amongst the first to show that the Beta variant was indeed highly resistant to neutralization. What we did was to take 44 individuals who were infected in the first wave of infection in South Africa, and therefore infected with the original variant, because this was before Beta had emerged. These 44 samples included a range of titers. You can see the titers are shown here down here, and you can see that the titers range somewhere from about 1 in 100 down here at the bottom, all the way up to 1 in 10,000, with titers in 1 in 10,000 characteristic of extremely ill hospitalized individuals. However, when we tested these 44 sera against beta, which had dominated the second wave, you can see that there's a substantial reduction in neutralization, and that's summarized by the orange line here, but really a ninefold reduction in titers. And perhaps even more alarmingly is that 48% of these sera showed no residual activity, neutralization activity of beta. So both plasma and in data that I'm not showing here, monoclonal antibodies showed significantly reduced efficacy against beta. This has subsequently been confirmed for beta and for many, many other variants. And I think there's been a lot of energy put into, correctly put into understanding immune evasion by these variants of concern and variants of interest, things that might come to dominate. But the thing is, we wanted to understand a slightly different question in our follow-up research, which is what about the immune response triggered by the variants of concern? In other words, how does the immune system differently see the variants of concern compared to the original ancestral variants? And if there are differences, can we leverage that information for vaccine design? We started to ask this question by simply saying, how good is the immune response to beta? It contains many of these escape mutations. How robustly does the immune system see beta? And is it less well recognized compared to the original variant? To do this, we worked with collaborators across South Africa, as you can see from this map, to study a cohort of nearly 90 hospitalized individuals who were recruited in January and February of 2021, at which point beta entirely dominated the epidemic in South Africa. These patients were recruited at Groote Schuur Hospital, which is shown down here at the bottom, right at the base of Table Mountain, in a cohort that had been established by Ntobeko Ntuzi and Wendy Burgers, and with support from Carolyn Williamson. We also worked closely with Tulio de Oliveira, whose lab performed much of the sequencing. And then we compared these 89 individuals with a cohort of very similar people, also hospitalized, who had been recruited during the first wave of infection, and therefore prior to the emergence of beta. This was a cohort led by Teresa Rousseau and Michael Boswell, and then here you can see my team. So what essentially we did was to compare immune responses in these two cohorts. And the major finding of this research, which I think was the first time that we began to be intrigued in the differences in how the immune system sees different variants, is that NAbs, or neutralizing antibodies to beta, appear to be more cross-reactive than those that had originally been triggered by the original variant. So that's shown here. This is the data that I showed you a few slides back. These were looking at the 44 individuals who had been infected in the first wave and infected with the original variant. What we did now was to take 57 individuals who had been infected with the beta variant in the second wave and ask the reverse question. So you'll notice here that the order of the axes is swapped. On the left-hand side, it's original and then beta. And on the panel here that I'm looking at now, you can see that we are first measuring responses to beta and then to the original variant. But what you can see is that although we have the same range of titers against beta, in other words, the virus that infected these people, we have much lower, slightly lower titers against the original variant, but much less of a reduction. So it's only a threefold reduction in titers compared to the ninefold reduction we'd seen with individuals in the first wave. And only 7% of these plasma showed no activity against the original variant. I think this was the first indication that that the immune response to spikes that vary in some cases only by a few amino acids may be fundamentally different. This was not true just of the original variants. We also tested a subset of these plasma from people infected with beta against the variant called gamma, or previously known as P1 from Brazil. And you can see that in this case, in fact, we saw a slight increase in titers, and all of these sera were able to maintain neutralization of gamma. So neutralizing antibodies to beta do appear to be more cross-reactive. We wanted to confirm whether this finding was true in a second cohort, and so we turned to a slightly different cohort of this time healthcare workers, also from the Groote Schuur Hospital, but a slightly different group of people. We compared here through three groups of healthcare workers. 20 of them had experienced no prior infection and then received the Johnson & Johnson vaccine, which was at that time being rolled out across healthcare workers in South Africa. They received the vaccine, and then we took their blood at 28 days post-vaccination. We compared that group of individuals who received only the vaccine to two groups of individuals who had been infected either in the first wave with the original variant and then vaccinated, or with 20 individuals who'd been infected in the second wave with the beta variant and then vaccinated. So three groups, all vaccinated, 20 of whom had no experience of prior infection, and we did this very carefully, and then two groups that had been infected either with the original variant or with the beta variant. The first thing, I think I've put this slide in here because I think it's good news. It's consistent with several other studies that have looked at what happens in people who were previously infected and then received various RNA vaccines, though this is the first data to my knowledge of looking at what happens with an adenoviral vaccine. But what you can see here is that pre-seropositivity, in other words, prior infection, results in massive boosting of neutralizing antibodies that are triggered by the adeno 26 Johnson & Johnson vaccine. So that's shown here at the bottom. You have the three sets broken into pre-vaccination and post-vaccination. Black is no prior infection, grey people infected with D614G, and blue people infected with beta. And you can see that with one exception, the titers in people who receive the vaccine only are fairly low. But in contrast, if you look at either of the two groups who were previously infected, you see massively increased neutralization titers, up in the order of 1,000 to 10,000. I think this is important data for South Africa, where we have, in many cases, huge levels of prior infection. In several areas, we have up to 40% of our population that has been previously infected. And in the context of an adeno vaccine that is a single-dose vaccine, the Johnson & Johnson vaccine, I think this suggests that we may get very good protection in these individuals who have previously been infected. But what about the immune response to these two variants? Again, that is the key question that we wanted to ask, is do they differ from one another? And it turns out that indeed cross-reactivity is variable depending on the spike sequence. So here I have the individuals at 28 days post-infection who received only the vaccine, so no prior infection. You can see that the geometric mean titer against the original variant is 74. But when you test against beta, we've previously published this, you see almost no residual neutralization. Against delta, 3 individuals did show some neutralization, but there's a huge knockout in neutralization. And again, this is consistent with others. This is what happens in individuals who were infected with D614G, the original variant, and then vaccinated with a vaccine that is based on the Wuhan 1 spike. So these two spike sequences differ from one another only by one amino acid, the D614G. You can see here that in people previously infected and then with the original variant and then vaccinated, massively high titers, 1 in 3,000 nearly. And you can see that they're able to maintain neutralization at extremely good titers of 1 in 600, even of the highly neutralization resistant beta and of delta. So very good cross-reactivity. The situation is different, interestingly, and you can see just straight away, look at the slope of the curves. The situation is slightly different in people who are infected with beta and then vaccinated with Wuhan 1. Here again, you see very high titers against D614G, very high titers, more than 1 in 1,000 against beta, so that matches the spike that they were infected with. But against delta, a spike that they haven't seen, you can see there's a significant drop-off in titers, a 6-fold reduction between beta and delta, very different to what happened in this group where titers were almost identical. So antibodies that are triggered by the original variant and by beta neutralize delta poorly. The reason that I think this is of importance for South Africa is that our subsequent third wave, which we're now in the middle of, is no longer beta. In fact, our epidemic is now entirely dominated by delta, as for many other countries across the world. And so I think that this does have implications both for reinfection, the possibility that people who are infected with beta, who assumed they would have been protected, may turn out to be substantially less protected than they hoped when they meet delta. But it also has implications, I think, for second-generation spikes, many of which now incorporate the beta spike. And I think we'll have to watch with interest to see what the level of immunogenicity triggered by those spikes is against both the original and delta and beta and all the many other variants of concern that are emerging. So the conclusions are that as for other vaccines, SARS-CoV-2 infection before vaccination with Johnson & Johnson significantly boosts neutralizing antibodies. I think in high SARS-CoV-2 serum prevalence areas, that includes South Africa, that includes many other parts of the world, this will significantly impact vaccine efficacy in a context of a single dose, low-cost, easy-to-store vaccine that is the Johnson & Johnson vaccine. However, all spikes are not equal in their capacity to trigger cross-reactive antibodies. This has implications, as I've already mentioned, for second-generation vaccines, these adaptive vaccines that now seek to include the spike of the beta variant either alone or in conjunction with others. We will need to isolate monoclonal antibodies from infections caused by variants of concern to understand the differences between the epitope targets, and I think this will be important for understanding therapeutics. And as population sweeps occur genetically in terms of the virus, I think that will also result in population sweeps in immunity that are likely occurring in the high context of high levels of infections by variants of concern. And this will be something else that we will have to watch at a population level. I'll stop there and thank our many collaborators. At the NICD, which is where I'm physically based, this is our team in the photo here, and you can see it's a team, a large team that they've worked tirelessly since the emergence of SARS-CoV-2 in South Africa, and I thank them for all their commitment. At the University of Pretoria, Teresa Rousseau, Michael Boswell, and Veronica Erkerman. Wendy, Ntbeko, and Carolyn have been just the most amazing collaborators over the last 3 or 4 months, and I thank them for that. At the University of KwaZulu-Natal, Tulio's team has been really wonderful. We in South Africa have a consortium. It was called the 501YV2 Consortium, should probably now be called the Beta Consortium or the Delta Consortium, but essentially a group of South African scientists who meet weekly to define what the major questions are, and I thank them for their input. Our funders, and then most importantly, the clinical teams and the participants. Thank you.
Thank you, Penny. This was outstanding lecture, and thank you for joining to this GVN meeting from South Africa. Muchas gracias, doctora Penny Moore, por esta brillante revisión con todo el impacto que tienen las variantes entre nosotros. Yo creo que es toda una enseñanza que se ha hecho las investigaciones en Sudáfrica. Debo pasar al siguiente conferencista en respecto al tiempo que tenemos. Permítame presentar al doctor Myron Levin. Venimos ya de Sudáfrica a las Américas. El doctor Myron Levin es un líder de investigaciones en vacunas. Él es un infectólogo pediatra, pero además un infectólogo en general muy reconocido, profesor principal en la Universidad de Maryland, donde él creó un centro o desarrolló un centro de vacunas. Él, además de eso, es un líder que ha trabajado muchos años en Sudamérica, especialmente en Chile y en el Perú. Anoche conversando con varias personas como hace 30 años el doctor Mike Levin presentaba una conferencia inolvidable sobre cólera en el año 91, cuando comenzaba la epidemia de cólera en las Américas después de 100 años. Además de eso, el doctor Levin ha sido líder tanto en la Sociedad Norteamericana de Enfermedades Infecciosas como en la ASTMH, o sea, la Sociedad Norteamericana de Medicina Tropical. Él nos va a presentar una puesta al día acerca de vacunas contra el COVID tan importantes a reflexionar entre nosotros, ya que en América Latina tenemos diversas vacunas en cada país, en cada región tenemos diversas experiencias de vacunación masiva con estas vacunas, así que sin más, quiero invitar al doctor Mike Levin, recordando que hace 30 años él fue uno de los que dieron las conferencias plenarias en Lima, Perú, en el Congreso Panamericano. Mike, bienvenido. Muchas gracias de estar con nosotros en este simposio de GVN. Doctor Levin.
Buenos días, colegas. Me llamo Mike Levine. Voy a dar una charla sobre vacunas contra COVID-19. Y en esta charla vamos a discutir que al comienzo de 2020, con urgencia frente al virus pandémico, tenemos que desarrollar vacunas para prevenir la enfermedad. Dos avances científicos básicos clave ayudó en el desarrollo de buenas vacunas. Uno incluyó el diseño de una proteína spike que mantiene su configuración pre-fusión. Esta configuración aumenta la poder neutralizante de anticuerpos estimulada por la vacuna. Segundo avance fue la adaptación de la tecnología de ARN mensajero con lipid nanoparticles para desarrollar vacunas contra el virus SARS-CoV-2. Hemos visto además varias otras vacunas. Una preocupación al comienzo del desarrollo de vacunas fue la posibilidad teórica que alguien vacunado con unos tipos de vacunas, si se infectarían con el virus virulento, ellos posiblemente, en teoría, desarrollarían enfermedad más severa que no vacunados. Por fortuna, esta situación no hemos visto hasta la fecha. Hemos visto una rapidez increíble de los ensayos clínicos utilizando diseños adaptables con una mezcla de fase 1 con fase 2 o fase 2 con fase 3 tipos de ensayos, otros fase 1 a fase 3. Increíble cómo rápido pasaron los ensayos clínicos. Las agencias regulatorias del mundo han trabajado juntos. El FDA, el EMA, MHR, colaborando con el OMS. Fue excelente ver la colaboración. Y ellos indicaron al comienzo que una vacuna con una eficacia de 50%, no más, con una eficacia del intervalo bajo de 30%, sería suficiente recibir una autorización para uso de emergencia. Como saben, las mejores vacunas fueron mucho más eficaces que eso, pero fue excelente ver la colaboración de las agencias. Después de la administración de millones de dosis con unas vacunas, hemos visto unos señales de problemas de seguridad. Vamos a discutir una reacción adversa muy rara, pero severa. Hasta la fecha tenemos problemas de suministro de vacunas a nivel mundial. Hay países con mucha cobertura, hay países como varios en África con poco cobertura de vacunas por falta de dosis. En todos los países del mundo hemos visto vacilación de un subgrupo de la comunidad a vacunarse y un subgrupo de ellos son completamente anti-vacuna. Tenemos que aprender cómo desarmar estos grupos y convencerlos a vacunarse. Hemos visto variantes de preocupación de mucho interés y son importantes en la evaluación continua de las vacunas. Y voy a discutir qué sería una vacuna ideal contra COVID-19. ¿Qué serían las características de una vacuna ideal? La vacuna sería segura en personas de cualquier edad, incluyendo huéspedes inmunocomprometidos y mujeres embarazadas y madres lactantes. La eficacia alta veríamos en todas las edades y en grupos de alto riesgo. La protección directa estimulada por la vacuna empezaría en pocos días y continuaría por años. Y la protección directa sería estimulado por una sola dosis que no necesitaría una aguja o jeringue, como por ejemplo por vía oral, por vía nariz, por la piel, sería posible vacunar. Las vacunas serían termoestables, no necesitarían una cadena de frío. Y personas vacunadas, cuando ellos encuentran el virus virulento, el virus no quedaría en la nariz. Como una cobertura alta en la comunidad, la vacuna ideal daría protección indirecta para proteger aún subgrupos no vacunados. La vacuna ideal sería posible fabricar en una manera económica a gran escala, producir un montón de dosis. La vacuna sería efectiva contra variantes de preocupación. Hemos visto cómo rápido preparamos varias vacunas candidatas. Hemos evaluado en ensayos clínicos rápido las vacunas. Hemos visto autorizaciones para uso de emergencia para varias vacunas. Muy buenos resultados. Sin embargo, sobre suministro, entrega y esfuerzos proactivos para minimizar resistencia al concepto de vacunación de unos en la comunidad, tenemos que mejorar la situación. Al comienzo de enero, la OMS notó más de 200 vacunas candidatas, entre ellas solamente 15 en ensayos fase 3. Entre los 15 en fase 3 en enero, 2 de mRNA, 2 de adenovirus y otras virus que no replican en el humano. 6 vacunas de virus inactivado, 2 de proteína subunidad, 1 de vacuna de ADN. Los primeros datos de vacunas en ensayos de fase 3 daron muy alta eficacia, evidencia de alta eficacia, incluyendo estudios hechos en Latinoamérica. Menos con la vacuna AstraZeneca, pero este es nivel de eficacia contra cualquier severidad esta vacuna funcionó bien contra prevenir hospitalización y muerte. ¿Qué han logrado las mejores vacunas? Alto nivel de protección, particularmente contra enfermedad severa. Al comienzo, protección de contactos domiciliares y contactos cercanos. Unas vacunas, la mayoría de investigadores aceptan que hay un correlativo inmunológico de protección. Estos son, por ejemplo, las vacunas de mRNA y también unos de proteínas purificados con adjuvante. Y el correlativo es el título de anticuerpos neutralizantes contra el virus salvaje. Las mejoras de estas vacunas protegen contra variantes de preocupación. ¿Qué más necesitamos? Evidencia de eficacia en niños, identificación de correlativos inmunológicos para cada categoría de vacuna y respuesta a la pregunta, ¿necesitamos dar una dosis de refuerzo? Si la respuesta es sí, ¿cuáles vacunas, cuándo y con cuáles antígenos en la vacuna? Mencioné variantes de preocupación. Hay más o menos dos tipos de punto de vista fenotípico. Hay unos como beta y gamma de Brasil, que causan una enfermedad más severa y necesitan mucho más anticuerpos neutralizantes neutralizar el virus. En común, estos variantes poseen una mutación E484K que afecta la eficacia de unas vacunas. Delta es otra cosa. El problema al nivel salud pública con Delta es su transmisión increíble. De recién, la semana pasada, publicaron en el New England Journal la protección de Pfizer y de AstraZeneca en Inglaterra contra Delta y contra Alfa variante después de una dosis o después de dos dosis. Una dosis, ambos, AstraZeneca y Pfizer, proveyó más o menos 50% de protección contra alfa, menos contra delta. Con dos dosis, la Pfizer es más o menos 10% más eficaz que AstraZeneca, de ambos buenos contra enfermedad severa. Buenas noticias. Y en Chile, hace unos meses, el Ministerio de Salud de Chile publicó en una conferencia un estudio que hicieron ver la eficacia de la vacuna Coronavac o Sinovac que compraron para la población de Chile. Y los datos son bien interesantes. Estos son datos muy preliminares. Si en individuos que habían recibido dos dosis de vacuna, la protección por Sinovac fue 85 contra hospitalización, 89 contra entrar una unidad de cuidado intensivo, 80% contra muerte. Hace unas semanas publicaron en el New England Journal los resultados. Estos datos son en individuos 60 años y mayor, y vemos muy buena eficacia con esta vacuna que no estimula altos títulos de anticuerpos neutralizantes. El señal de seguridad que hemos visto en Europa y en Norteamérica fue el síndrome de trombosis en el cavernous sinus del cerebro o en venas abdominal profundo con trombocitopenia y con títulos elevados anticuerpos a platelets. Son raros, pero severas. Puede ser letal. Ocurre más o menos 6 a 19 días después de la primera dosis en adultos entre más o menos 20 y 50 años de edad, mujeres más que hombres, y como dije, la mayoría en Europa y Norteamérica. Otro señal fue Bell's palsy con unas vacunas, pero los datos son en conflicto del punto de vista de la fuerza de la asociación. El problema que encontramos como un planeta, una población mundial, es la diferencia en vacunación de países ricos y países intermedios con países con pocos recursos. Tenemos que resolver este problema. También mencioné vaccine hesitancy, muy difícil cambiar la actitud de gentes. En resumen, las primeras vacunas de varios tipos, a pesar de problemas al comienzo de provisiones para preparar las vacunas, lo prepararon, hicieron buenos estudios y mostraron buena eficacia. Al comienzo vemos problemas en la entrega de vacuna. Por ejemplo, Pfizer, que necesitó al comienzo menos 70 grados en su cadena de frío. Podían resolver este problema. Mencionamos el grupo antivacuna con su desinformación en el Internet. Es un problema global. La inequidad discutimos y las variantes de preocupación que no fue previsto al comienzo de la pandemia. Y gracias por su atención.
Muchas gracias, doctor Mike Levin. Como siempre, una revisión exhaustiva y muy rápida de la importancia del tipo de vacunas que necesitamos, y le agradecemos por participar en el Symposium GVN. Vamos a pasar al último conferencista, el doctor David Friedman, que se entrenó inicialmente en la Universidad de McGill, Canadá, su tierra natal. Desarrolló diversas investigaciones en Brasil, Perú, en varios países africanos, y además de eso mantenía su posición de profesor de enfermedades infecciosas de la Universidad de Alabama, donde actualmente es profesor emérito de esa universidad. Además de eso, participa en el grupo clínico de la Sociedad Norteamericana de Medicina Tropical y es muy distinguido por haber trabajado en una red internacional de medicina de viajeros. Y desde ayer, además, es miembro honorario de la API, ha sido miembro honorario de la Universidad Peruana Cayetano Heredia, donde él ha dirigido el curso Gorgas International Clinical Tropical Medicine. El doctor Friedman nos va a presentar the international travel and COVID vaccine. David, welcome. Bienvenido.
No.
Favor, el micro para el podio del doctor Friedman.
Okay, there we go. Okay, so I'm going to talk about some practical issues wearing my travel medicine hat. These are not really scientific issues, but these are everyday issues that I think are going to affect not only us, but the people we advise. Those are my disclosures again. The world's changed. I used to run a travel clinic. It had to close for over a year, and I found myself standing in the parking lot at the stadium giving vaccinations there instead of in the clinic. So I'll talk about some nuts and bolts about entry requirements related to vaccines, not really about testing requirements, and a few practical issues, especially in people that are in a rush to travel, or you need to know whether they're prepared or not with an acceptable vaccination regimen before they travel, what documentation, and I'll finish with a little bit about digital health passes. Essentially every country in the world has some kind of travel restrictions or entry restrictions, and there's only about 4 or 5 countries, including a couple in Latin America, that have absolutely no restrictions. I've put up the example of our host country here, the Dominican Republic. All I can say is these get longer and more complicated every day to interpret. In terms of safe travel to a country or hosting as a tourist receiving country like the Dominican Republic I showed this earlier, but these are these are a choice of sort of major destinations or well-vaccinated countries. I've highlighted a couple in Uruguay and Chile, and you know, in general, right now, in fact, I mean, they're big areas, but the European Union and the United States are high, but they're not. They're certainly not the highest of the regions of the world now. I am actually going on vacation in September to Iceland because it was one of the reasons it was nicely at the top of the list. So this is just an example. One of the things that I do now with people that I work with now in the private sector is we keep track of these things and you know, host an information source. So there's 133 countries now where vaccination will help you. There's only a couple of countries, as far as visitors or tourists or short-term people are concerned. There's actually only 2 countries where they deny you must be vaccinated in order to enter. In most of the countries, 133, that have some sort of requirement, it gets you some exemptions, it gets you some considerations. You know, if you're vaccinated, you don't have to travel. You can go there if you're from a country that they accept people from. You can go there, but you can't, you may have to do something extraordinary that would make it impractical to go there for a short trip. If you have to quarantine for 2 weeks, obviously, you know, you're not going to go on vacation there. So that's the list of countries. Usually you get exempted from one of the things that are shown there, either having to take a test before you leave, a test on arrival, quarantining, etc. So that's the list. One of the tricky practical issues is what's an acceptable vaccination, and I know this is controversial in many parts of the world. Most countries do list what they require and what they accept, and you do need to check that. Oftentimes they're using a number of sources to make their list. Some very sophisticated countries may make up, really look at the data, for every vaccine. But in general, countries, say, okay, we accept all FDA-approved vaccines, we accept all WHO-approved vaccines, we accept European EMA vaccines. That's important because the EMA says you must, to go to Europe, you must get one of the ones that we approve, but it's important to know that a lot of countries have broken away from that. So 15 or more of the European countries will accept people that have had a non-European approved vaccine. And then the big controversy is the Chinese and the Russian vaccine as to which countries accept those. And so I've broken those out for you, and it's getting better. An increasing number of countries will accept the Chinese and the Russian vaccine. Some countries are making a distinction between the AstraZeneca vaccine made in Europe and the United States and the version of the same vaccine that's made in India called Covishield. And that's been a little bit of controversy because, again, technically, the EMA has not approved Covishield, but most, as I said, the majority of European countries are accepting now the Covishield, but not all. So you do need to check. So this list is, and in general, they have different rules, okay, as to what a fully vaccinated person might be. And here are the variations. To simplify it, though, 66% of those 133 countries say finished your series 2 weeks prior to arrival. And then the next most common is any time prior to arrival. They don't specify. They just say, you've had 2 doses of vaccine, you're fine, or 1 dose of Janssen, you're fine. But then there are some countries that have very complicated things, and I've listed those there. It's not too many countries, but you may run into some of those complicated requirements, or they say your vaccine's only good for 8 months or 9 months or something like that. So occasionally you'll run into something like that. This is non-vaccine related for travelers, but just sort of a look as to what arrival measures you may run into. And in fact, there are 200 countries now that, at least for some travelers, have pre-departure testing. So sometime before your flight to that country, you need to have some kind of test, and again, they specify whether it's PCR, antigen, what it is, and then you see the various quarantine. Quarantine options that are there. I've not included all the possible options. There are some outliers here. For example, Hong Kong right now has a 21-day mandatory quarantine. So there are some countries that are much more severe. Israel is implementing, again, for most countries, a 14-day, even for vaccinated people, a 14-day mandatory quarantine. So those are out there. Just as a comment, from a safety point of view, I think if you're doing testing, there's no reason to have more than a 7-day quarantine. If you're testing before the flight and then testing 4 or 5 days after the flight, as well as on arrival, I really don't think it's necessary to have more than a 7-day. We've talked a little bit in the earlier session, for those that were there, about the prime boost strategy. So, the fact that an adenovirus vaccine followed by an mRNA vaccine seems to be the best sort of thing. One of the problems we're running into is that if you mix your vaccines, though, right now, the regulations say that many countries will not accept a mixed series, and that they just say it has to be 2 Pfizer doses, it has to be 2 Moderna doses. So if you do one of these options, the country may not accept you as being fully vaccinated. I think this is probably going to change moving forward, but that's a real problem, and many people from countries that have implemented as a matter of policy mixed vaccine series, and that includes Canada and a number of the Western European countries, allow mixed series, and a lot of other countries will not recognize right now those people as being fully vaccinated. As far as other combinations are concerned, and especially in terms of making sure your travelers are safe, you know, there are certainly, we don't have data, there's nothing published, but anecdotally, you know, many people who've gotten Chinese or Russian vaccines have gone to the United States or Europe and gotten booster doses with RNA vaccines especially, and we don't know of any problems. No problems have been reported, you know, and you know, the immune response is probably good. And, you know, from an effectiveness point of view, if people ask me, I think we would all say that at least one dose, if you've had a Chinese or a Russian vaccine, you should probably receive at least one dose of an RNA vaccine to be sure that you're protected. Whether you need 2 doses after, you know, an inactivated vaccine, I'm not sure. There's no data one way or the other. And of course, don't forget that if you've had a previous infection, we're going to respond much better to one dose anyways. I showed this slide earlier, and again to show on the right, you know, the best off people, your most protected travelers are clearly the people that have finished a 2-dose vaccination, or if they've had prior COVID, 1 dose is fine. So if you're introducing vaccine and your traveler says, you know, I want to go to Western Europe, I want to go to the United States where things are bad, if you know for sure that they've had prior COVID, and you have their test results, et cetera, they should be good to go after 1 dose of vaccine. From a protection point of view, although if the country has an entry requirement for vaccination, they may need to get 2 and not be ready to travel. Okay, let's go to the last topic, which is documentation and digital passes. So the WHO is recommending that it be documented on one of these antique little yellow books, little yellow cards here. You know, I don't even know if a lot of countries, you know, are recognizing this, or all the border guards would know if you put a COVID vaccine on one of these, but that's the official WHO recommendation. Whatever else you do, you know, make a note of it in the ICVP as well. Many countries have different kinds of evidence. This is the CDC card from the US. You know, right now, I mean, they're very easy to forge. There's a lot of fake cards out there, but just about any receiving country, if you have one of these, you know, white cards, or you have some official looking piece of paper from your home country that's marked with the name of a national authority, right now, just about every country, you know, will accept it. As long as you can convince the border guard, you know, that you've been vaccinated, that's really all you need. I think this is going to change. Takes a while for mechanisms to get in place. What about digital? Well, the one that seems to be in front right now in terms of implementation is the European Union. They have the so-called digital green pass, which is issued mostly for travel within Europe. Although, you know, I think some other countries would recognize that as proof of vaccination. The problem, and you can use this app either to show that you have a recent negative test, or if that's a requirement, or that you've been properly vaccinated, and it actually keeps track the day of your test and how many hours it's been since you took your negative test. You get one of these QR codes. The problem is that the countries have all been told that they can issue certificates, a digital pass to foreigners. It has to be issued by the first country in Europe that they're arriving in. Honestly, it's not a priority for most of the countries, so most countries in Europe have no mechanism yet to issue these to foreigners. What you actually need to do now, if you're in places like France, Germany, whatever, if you're going to a museum or a place that requires you to have proof, then you can go to a pharmacy or you can go to a lab and get an antigen test or a PCR, and they'll give you a piece of paper, and usually in that country it's good for 2 or 3 days, you know, and you can go into public places, but you know, if you're there for several weeks, you need to get a test an antigen test every 2 days or a PCR every 3 days. So that's what's happening now. That's what's happening now in Europe. And there's the list of countries. It's getting longer every day. The majority of the EU countries now require a health pass to enter indoor public places. Worldwide, unfortunately, it's a very fragmented environment. IATA, the International Air Transport Association, the Airlines Association, have developed the IATA Travel Pass, and they're testing it with quite a few major airlines. There are private companies, Clear, AOK Pass, IBM, etc. Everybody's developing a different app, and that's part of the problem. And I think what there's a lot of issue about standards, and this initiative here, the Good Health Pass, is trying to initiate standards. WHO, ECDC, they all have different sets of standards, and everything needs to operate together. The border, the airlines, the labs, the vaccine centers, and that's a real technical hurdle right now, and I don't know who's going to dominate. We're going to have to see how this works out, but right now, by and large, we're in a situation where a lot of airlines have their own documentation you have to upload. A lot of countries, you have to go on some kind of national border website before your trip and upload your information. And I think for now we're stuck with that, except maybe for Europe, within Europe, and for Europeans, that if you go to a country, you're going to have to upload all your stuff maybe twice, once for the airline and once for the border authorities in the country. It's just going to be a while before we have a real international certification. Maybe the EU will dominate. I don't know. The EU won. Considerations, as I said, there's a very complicated set of rules. There's a number of websites you can use to find out various levels of detail on what the considerations are. I'll finish with, just interpretation of certain requirements, and this is still causing a lot of confusion. So the proof, a lot of countries will give you better conditions if you have a proof of COVID-19 recovery, and there are various rules on what proof of COVID-19 recovery is, how long it lasts for. Usually you need to have the record of a positive test result at the time that you were ill. A few countries accept an antibody result, but it's kind of very confusing. The second one there is, again, in the U.S. we have a lot of home tests, as they do in many countries. Does the country accept a home test? In the U.S. you need to do it under video observation, and there's some companies that offer that, a telehealth observer. And so that's something to keep in mind. And then the rules about direct flights and indirect flights can be complicated. Some countries say you need the test 72 hours before you arrive in the destination. Some say within 72 hours of leaving your home country. Some say if you're taking a connecting flight, it needs to be 72 hours before your last flight to the destination country leaves. So those are, nuances that cause a lot of confusion. And then the issue is, I've shown in green there, 3 days before departure, 72 hours prior to departure. Not the same thing. So, for example, it's 70, you have to figure out what time your flight is leaving, and it has to be within 72 hours of the departure time of the flight. What's going to happen if the flight's delayed 2 or 3 hours and you're close? Hard to know. But the United States, and this causes a huge amount of confusion even amongst the travelers, even amongst the home test. I did a home test on Wednesday before I left here because I'm going home on Saturday, and it's good for 3 days. So the rule in the United States is 3 days, very flexible. If you're coming home anytime Saturday, as long as you've had a test anytime Wednesday, Thursday, Friday, it can be a little bit more than 72 hours. Other countries have it different, but I got in an argument with the person on the phone because they thought it was 72 hours while I was doing the home test. And right now in the U.S. anyways, and these are big companies, so these are the two that meet U.S. re-entry criteria. So they give you an app, and you log on, and somebody watches you. They check. You have to pre-register with your passport and everything like that. They check your identity. You have to show your passport to your phone, and then they watch you stick the thing in your nose and make sure you turn it 3 times, and then they look at the result. You show them the result, and then they verify it and issue you a certificate. So maybe these are going to be a little more popular. Practical issue, these are antigen tests. Even with a 98% specificity, there's going to be false positives. And if your country has a very low prevalence at the time you do this, your home country, then the majority of tests are going to be a false positive because there are very few true positives in your country. So you're going to get quite a few people getting false positive results. And so, even the company advises it, don't do it 1 day before, even though it's a self-test and you get an answer in 15 minutes. Do it 3 days before, because if your test is positive, you can then you have enough time to go and get a PCR somewhere. A little less convenient, but you can get a PCR somewhere to figure out if it was a false positive or not. So these are all practical issues we're working out. The issue about traveling, I think the biggest issue now is vulnerable people. And, even vaccinated vulnerable people should not travel to high endemic areas. I mean, we don't know enough, especially about the duration in vulnerable people, and that it has to do with third doses and things we haven't sorted out. And this is a story of an infectious disease professor from New Jersey, and he had so many risk factors, but he was 81 years old, and he felt he had to go to India during the crisis there to help out his family. He had heart disease, he had diabetes, there was no PPE in India. Anyways, he got COVID in India and died, even though he was fully vaccinated. So, don't stress the system. As I said this morning, don't go to a crowded club, during the holidays, and don't go to a high-risk country, even if you're well vaccinated. So I think I'll stop there, and I guess we have no time for questions. So thank you very, very much.
Thank you very much, Dr. Friedman. Because we finish with this 4 lecture, and we need to move into the next symposium here. I would like to close and first renewal the GVN network to try to invite to all these leaders from virology in Latin America. Quisiera terminar esta actividad invitando a los líderes de virología de América Latina a que se unan y participen de la GVN Network. El doctor Christian Brehoff, que es el presidente realmente de la red, así como el doctor Robert Gallo, fundador de esta alianza estratégica importante, invita a todos. El doctor Gallo también dará una conferencia plenaria y quisiera agradecer a todos los conferencistas por esta excelente revisión que vino desde lo básico, mirando la inmunidad y pasando por todos estos conceptos importantes de las vacunas en los viajeros, etcétera. Rol que los infectólogos muchas veces participamos asesorando a nuestros pacientes, a nuestra población sobre el impacto de los viajeros, que representan en muchos de nuestros países una fuente importante de la economía a través del turismo. Así que yo creo que hemos cubierto estos 4 puntos. Agradecemos a la Asociación Panamericana que hayan permitido tener este simposio y a todos ustedes muchas gracias por su participación. Igual agradecer a los speakers por estas 4 charlas tan importantes de Italia, de Sudáfrica, de Estados Unidos y acá en el Congreso con presencia del doctor David Friedman. Muchas gracias y cierro este simposio. Muy amables.